Hello, AP Bio students or Miss Davis or random internet people watching this video out there. I'm Derek and I'm here to talk about mitosis. Oh yeah. What is it? So mitosis is pretty much just cell division. Creates two identical daughter cells from the original, and it occurs mainly in somatic or body tissue cells to replace damaged or dead ones. So pretty much the stages, you don't technically need to know this, but I always think it's helpful to know it, and I always use this little device that I learned. I passed my algebra test: interphase, prophase, metaphase, anaphase, telophase. I find that it works for me. If it works for you, great. So interphase, there's three subphases. There's G1, which is pretty much the cell just doing its thing, just normal cell stuff. It's synthesizing protein, it's growing, it's doing whatever needs to be done. And then it goes to the S phase, which the DNA synthesizes, it replicates itself, and does that so that in the end the daughter cells have a full set, or diploid, they have a full set of chromosomes. And if they don't, that's not good. So in S phase, DNA replicates itself, and it goes to G2, which goes back to doing normal cell stuff. And then also the organelles replicate to get ready for mitosis. So prophase. In prophase, the chromosomes condense, so if you look through a microscope, you can actually see them. And if you're looking through a microscope, the nucleolus would be gone. So, and then metaphase comes next. So, metaphase is interesting because the chromosomes line up in the middle, like this, along the metaphase plate, which isn't actually a plate, just kind of an imaginary line. And then that's caused by like the pulling of these microtubules. It's like if it were a perfectly even like tug of war. They move back and forth, but eventually they set in the middle. Alright, so anaphase. I think this was a really cool picture. I don't know why. It's just cool. So in anaphase, pretty much the chromosomes are pulled apart. You know, the protein that was holding the chromosomes together splits, and the microtubules are pulling them apart. So, yeah. And then next comes telophase, which is reforming in the nuclear envelope, so you have pretty much two nuclei in one cell right now. And then cytokinesis comes up, and cytokinesis pretty much is the process of the cell cytoplasm replicating, and then the cell actually like breaking off into two now. And that process is called cleavage, and there's a little cleavage furrow, which is like this little gap that I'm doing here with my hands, and then eventually they separate. So mitosis control. Obviously we need to regulate when our cells go through mitosis and when they don't. So there's checkpoints, which are stoplights. You know, they said, okay, we can either keep going with this or no, like abort, abandon ship. You know, something's not right here. And then we can do that with cyclin. And so cyclin is a protein that builds up during interphase. And then that combines with cyclin-dependent kinase, and those two combine to form MPF. And MPF phosphorylates other proteins, and those proteins phosphorylate other proteins, and MPF pretty much activates this waterfall, this cascade of events that start mitosis. And it's cool because after like a certain amount of time, the cyclin in MPF automatically starts to break down, and then so it shuts off again by itself. Or it should in normal cells, but sometimes it doesn't. But that's kind of cool, like one like automatic self-check that the cell has to make sure it doesn't go crazy. And this little squiggly thing here, that's actually one of the cyclin-dependent kinases. That's actually like the 3D structure of it. I don't know, I just thought that was really cool to look at. Kind of like a ribbon gone wrong. And then cancer is mitosis gone bad. You can see my picture of little cancer cells down there. Oh, I got a mouse right here. And then it comes from the loss of control, like maybe those checkpoints aren't working, or cyclin isn't breaking down at the end of mitosis. So something just makes the cell just keep like just divides nonstop, and sometimes it divides incorrectly, and that's how we end up with like the really bad cancer and how it just like spreads everywhere. And so this is pretty much the end of this video. There's a sequel about meiosis. Check it out. So peace out. Oh.